Okay, so good morning, everybody. Sorry for the change in program this week. Maybe I'll tell you a little bit about it next week. I had a bit of an injury this week, so I had to kind of modify my work here. Anyway, I hope everyone had a chance to review the lecture recording on topic 13 and talked a little bit about DNA structure, a little bit about the discovery of the DNA structure, Watson and Crick, and a few other historical experiments, Frederick Griffiths, and we're going to address that in today's lab. So topic 13 was important kind of as a touchstone to get us into the rest of the topics. Particularly for today, we need to know a little bit about DNA structure. And so what we're going to do, just do a quick review on DNA structure at the beginning of this. But this particular unit is DNA replication and repair. And so today we're going to do the replication part. This is about a lecture and a half of material, so we'll carry some of topic 14 into next week. All right, so first, there is a review of DNA structure. And so if you take a look, this is, we call this the double helix. And the reason why it's called the double helix is because there are two strands. So if you take a look, I'm just looking at this picture in the middle. And here's one strand here. And here's the other strand there. And when the two strands go together, they kind of twist a little bit around each other. And so it's called the double helix. There's a couple of things to take a look at when we look at this structure. First of all, we have this phosphodiester backbone. So if you take a look here, we've got a sugar. So this is a sugar. This is a pentose sugar. It has 5 carbons. And we have a phosphate. And so it goes sugar, phosphate, sugar, phosphate, sugar, phosphate, and so on. And these phosphates have negative charges. That's important for gel electrophoresis. And then on the inside, these things here are nitrogenous bases. So I'll just write that down, nitrogenous bases. And the nitrogenous bases, of course, match one another. We call this complementary. So that means the A pairs with T, G pairs with C, and so on along the structure of the double helix. So this is the question, right? And this is, I've had this question on final exams, and usually it's there in some form or another, whether it's a multiple choice question or an actual written question. So it says, what does it mean that the two strands are complementary and anti-parallel? So let's take a look at that. So complementary basically refers to the base pairing. So those two strands are not identical, but they're complementary. So the base pairing, right? A pairs with T, so A is adenine, pairs with thymine. And if you take a look, between the two bases are these dotted lines. And if you remember way back at the beginning of the course, these are actually hydrogen bonds. So adenine and thymine pair with 2 hydrogen bonds each. G pairs with C. So guanine pairs with cytosine, and you can see we've got 1, 2, 3 hydrogen bonds. And so they're complementary. G complements C, A complements T. So that's what the complementary part means of the double helix. Anti-parallel. So anti-parallel, again, hopefully everyone reviewed lecture 13. And this is referring to the orientation of the chemical bonds. So I just want to draw for you deoxyribose here for a moment. So deoxyribose is a 5-carbon sugar, and it looks something like this. So this is just the sugar alone and not bonded. And so we've got, let me just think here. There's an OH here and an OH here. So that's what it looks like without being bonded to the nitrogenous bases or being bonded to the phosphates. And so when in chemistry, when we're looking at a compound, it's useful sometimes to number the carbons. And so if we number the carbons on this, as you can see in the top diagram, this is number 1, this is number 2, this is number 3, this is number 4, and this is number 5. And so sometimes we want to talk about, okay, the second carbon. So if we know what the numbers are, we can tell you which is the second carbon or the third carbon or the fifth carbon or whatever. Now, with DNA structure, we have the pentose sugars. We also have the nitrogenous bases. And so what if I'm talking about the second carbon? Are we talking about the second carbon on the nitrogenous base or the second carbon on the sugar? And so on and so on. So everyone decided that the nitrogenous bases are more important. And so the numbers on the pentoses, they decided to call them primes. So 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So the prime is that little tick beside the number. And so we have the two most important ones are the 5 prime carbon and 3 prime carbon. And the reason for that is that that is where things are connected. The 5 prime carbon is connected to the next nucleotide. The 3 prime carbon is connected to the previous one. So if you take a look at our strand here, sorry, I forgot that was going to take up the space, but we talk about the one strand of DNA going in the 5 prime to 3 prime direction. And the other strand goes in the opposite direction. And so this is just the chemical orientation. You can sort of think of it like reading. When we read in the English language, we go left to right. And so if we had every other line going in the opposite direction, then our lines would be anti-parallel. It's the same with DNA structure. We read all of our structures 5 prime to 3 prime. So you can see the strand on the left is going one direction, the strand on the right is going in the backwards direction. So we're going to talk a lot about 5 prime to 3 prime. So remember that. That's an important concept when we talk about replication, transcription, and translation. That 5 prime to 3 prime is the direction that nucleotides get read. So you can see the notes that popped up here on the left. It says here that enzymes can only add nucleotides to the 3 prime end. And that's something I'm going to talk about here in a minute or two. There's some arrows. I guess I need to draw some. Okay, so going back to Watson and Crick from topic 13, when they looked at the structure of DNA in their original publication, they actually had a sentence. And the sentence, you can read that at the top. It says, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." Watson and Crick. And so their thought was, and this was later proved, that all you had to do to replicate DNA was to separate the two strands, and then automatically you knew what the other strand was going to be if you knew this complementary system. So if you had an A, you would pair that with a T you would pair with an A C with a G, and so on and so on and so on. And it turns out that this is a huge part of the basis of DNA replication. So this is what we're going to talk about today, DNA replication. By the way, I guess I forgot to say at the beginning, I'm not sure how familiar everyone is with Zoom. If you do have a question along the way, you can unmute your microphone and ask a question. I don't mind being interrupted. The other thing you can do is there is a chat feature, and if you type something into the chat, it will pop up, and I can address that. I may not get to it immediately, but I will probably notice it relatively quickly. Okay, so let's talk about DNA replication. This is the first thing that happens is the two strands separate. And so this actually is the name. We call this the replication fork. So you can see there's the name there. There's a lot going on in this picture here, and we'll kind of get into this eventually here. But you can see that the DNA strand replicates, and we get two new daughter strands being formed. One of them is a leading strand, and one of them is a lagging strand. And we'll kind of get into that terminology here in a few minutes. But first, let's just take a look at the replication fork. So let's take a look in bacteria, first of all. So remember, bacteria have circular chromosomes. So you can see there's my circular chromosome. It's got two strands of DNA. It's a double helix. And DNA replication starts at something here called an origin of replication. So sometimes origin replication is abbreviated. It kind of looks like this. ORI. Sometimes they put a little R. in front of it like that. So I can't remember if you see something like that in my notes. I believe it is referred to that in the lab manual. But that's the starting point. So the origin replication, just for a definition, it's basically it's a sequence of DNA, so a chunk of DNA, and this is an indication to the enzymes where to start replication. So where we're going to begin copying our genome. So basically, that's where it separates, and you start getting these replication bubbles. And you're going to get 2 new strands of DNA. So you can see the new strands of DNA there are done in the lighter blue color. We'll call those the daughter strands. And basically, it's going to just separate a little bit more and a little bit more. The replication is going to move away from the origin replication. It's going to move in both directions. It's just more efficient that way to multitask and to move along in both directions. And that opening part, sometimes it's called a replication bubble. So you can see the term there. I actually have an electron micrograph. I'll show you of a replication bubble. So here we go. It's going to keep separating, and it's going to go all the way around and eventually go all the way around the entire chromosome, and you're going to get 2 daughter DNA molecules. There's the electron micrograph of the replication bubble. You can see the arrows there are where the replication is occurring, and like I said, it's just going to go all the way around until it eventually finishes the chromosome. So that's what happens in bacteria, circular chromosomes. Eukaryotes are a little different. I'll show you that in a moment. So just a reminder, when we talk about bacteria, they will often have plasmids. Not all bacteria have plasmids. Even E. coli sometimes has plasmids, sometimes it doesn't. But a plasmid is basically a very small chromosome. It's a mini chromosome. A normal chromosome is going to have 4,000 to 5,000 genes. A plasmid might have 2 to 5. But in order for it to replicate, it does need an origin replication. And so the plasmid that we extracted last week in the lab has an origin replication that's recognized by E. coli. In fact, the plasmid that we extracted is what we'd call an artificial plasmid. It was made by some scientists. And the main thing they did was made sure they had a DNA origin replication, or an E. coli origin replication, and then it doesn't matter what the rest of the sequence is. That's what will make it copyable by E. coli. Something else which is just kind of interesting, plasmids, they're not like big hoops. They, rather than, if I think of my big hoop, it sort of looks something like this. They actually pack themselves. So they sort of twist themselves up a little bit. And the main thing is to be able to pack everything in the cell. They have to wind themselves tightly. And so this actually is a term. It's called supercoiling. And I think it comes up somewhere in the lab manual. So I thought I would show you what that looks like. Okay, let's talk about eukaryotes and what they are doing. So they have way more DNA. So the human genome is the haploid genome is 3.2 billion base pairs, I believe. So, if you look at the whole diploid, that's around 6 billion base pairs, compared to E. coli, which is like 4 million, right? So what do we do about that? It's going to take forever to copy all that DNA. So we have multiple origins of replication. And so you can see there's our little genome, a chromosome. It's one of them anyway. Each chromosome is going to have its own and several of its own. And so it's going to make several replication bubbles. And like the last diagram, you can see the parental strand is the dark blue, and the daughter strand is the light blue. And we've got these little replication bubbles and these little replication forks. And eventually these bubbles are going to expand as the replication proceeds. And there's an electron micrograph. You can see a whole bunch of replication bubbles there. You can see there's one here, another one here, another one here. And the arrows represent the replication forks. So eventually, the replication bubbles are going to merge, and you'll end up with your 2 daughter DNA molecules. And that is, that's kind of the basics of replication. Okay, so, there's another electron micrograph for you. And you can see, again, a handful of replication bubbles. Okay, so let's get into some more nitty-gritty details of DNA replication. We need to cover a little bit of chemistry here. So if you take a look at this, here is a strand of DNA. And the thing that I want to point out is this here, this 3' end. So this 3' end has an OH on it, so a hydroxyl or an alcohol group. And that is actually the substrate of our DNA polymerase. So our DNA polymerase needs to bind there on the 3' end. And so this is why, as you can see up at the top, it says here, the DNA replication occurs at the 3' end. So 5' to 3', this is the direction of DNA replication. So something else to know is the other substrate is this thing here. So I'm hoping to have some space to write on the board here. But this thing here that I just introduced, we'll call it a DNTP. I'll define that in a second. A DNTP is a deoxy nucleotide triphosphate. So if you think about it, there's going to be 4 of them, right? So we're going to have DATP, we're going to have DTTP, we're going to have GTP, and DCTP. There we go. Because we have 4 different possible base pairs, right? So those 2 things have to get together. And if you take a look, that has 3 phosphates. So probably no surprise when you break one of those phosphate bonds, this releases energy. And so this is done by DNA polymerase. And then it's going to form this new phosphodiester bond here. You can see this, this double phosphate, that's actually called pyrophosphate, is being let go. So the energy from breaking down that nucleotide makes the new bond in your DNA. So I want to write down some of these notes, some of the things I just talked about here in a moment. I want to talk a little bit more about this enzyme, DNA polymerase, because this is the workhorse. This is the main enzyme that is going to do our DNA replication. So there's a lot to say about it because it's really it really does quite a bit, this enzyme. There's a picture of it. Like I said, I'll write some notes here in a moment. Just want to show you this nice model diagram from the textbook. You can see you've got the template strand is this purple DNA right here, and the new DNA there is that yellow strand. And there's actually a few different versions I'm not going to worry about in this class. You can see this one is specifically DNA polymerase 3. The main one is actually DNA polymerase 2, but don't worry about the numbers. It does refer to them in the textbook. I'm more concerned that you understand the principles of DNA replication. So as I mentioned, I wanted to make some notes about this. So the first note, as I've mentioned, is that DNA polymerase has 2 substrates. So let's write that down. So DNA polymerase has 2 substrates. So the first one is our new nucleotides. So remember, these are DNTPs. DNTPs. So we have A, T, C, and G. I'm not going to write out the DATP and so on like I did in the previous slide. The second substrate is our 3' OH end. So 3' OH end. And that's on the growing strand. Or if I can change my ink color. Let me just give me a second here, see if I can do that. Thought I could do that. Pointer options. There we go. Ink color. The red is just a little much, I think. Let's try this. I think this is purple. Okay, so that's the first thing to know. DNA polymerase has 2 substrates, right? And so as a consequence, okay, so I'll put therefore, it synthesizes our new DNA, synthesizes the new strand or the new DNA in the 5' to 3' direction. So like I said, you need to know that 5' to 3'. That's a really important concept. It's going to come up again and again over the next couple of units. Okay, and then the last thing to know, well, not the last thing, but the next thing to know, maybe the most also very important thing, is that the new strand is complementary to the template strand. Okay, so that's something we've already mentioned as well, and hopefully you know that. So let's write that down. The new strand is made complementary to the new strand. I guess we could call that the daughter strand. Is made complementary to the template strand, So remember that means A pairs with T and C pairs with G and so on. And it's worth putting that in a box. Okay, so that is not everything about DNA polymerase, unfortunately. But if you're going to learn anything, start there. Like I said, these are really important concepts, knowing that things are complementary, understanding this 5' to 3' thing, and the two substrates, right? And so it's really connecting the growing end, which is substrate number 2, with a new nucleotide, and that new nucleotide is going to follow these rules, these pairing rules. Okay, there's a couple more things to know about DNA polymerase, and so I'm just going to wipe this and write them down as well. If you don't get all this, by the way, I am recording the lecture, and I will be posting that a link for the recording once I get a chance to do so. Okay, a couple more things about DNA polymerase. So DNA polymerase, we'll call it DNA pol, unfortunately cannot start synthesis on its own. So it cannot start synthesis without that 3' end. So the consequence is it needs, and we'll talk about what this means in a minute. So it needs a 3' OH primer. So a primer, it kind of means a starter of something. And we'll talk about where that starter thing comes from in a bit, but it's actually going to be an RNA primer for some reason. Another thing that is interesting about DNA polymerase is it has proofreading ability. So DNA polymerase has proofreading ability. So I think it's a pretty good enzyme in general, and I'm trying to remember the stats on that. I think it makes a mistake about 1 in every 10,000 base pairs, but it also proofreads, and so that after proofreading, it's like 1 mistake in every 2 billion pairs. So that means it goes along and double checks its work. That's a really important feature if you think about it for this particular enzyme, because otherwise we'd have mutations all the time, and mutations are not usually good. They're usually very, very bad. So the last thing, and this will come up again a little bit later, is that the DNA polymerase, as part of its proofreading and part of the DNA synthesis, is it can remove our primers. So our primers are RNA primers. And so we'll talk about what that looks like here in a few minutes. So it can remove RNA primers. So that's my last note for now. So hopefully that's enough to kind of get us started on how DNA replication actually works. And like I said, this is a really important unit. And I'm going to come back to some of these concepts after the break as well. And hopefully the recording being available to you will also help to review this material. So let's take a look at DNA replication. Here's another image very similar to the one I showed you before. You can see you've got the 2 strands, you've got the template strand. So this is the one that is being looked at by the enzyme, and you've got the new strand, and it's growing right here at the 3' end. and you've got one of these DNTPs coming in, and this one is going to be complementary. You can see we've got an A, so the new one is a T because it's complementary to the A. So the DNA polymerase enzyme is going to grab that, bring it in. It's going to break down the DNTP, take off the two phosphates. There they go. Like I said, two phosphates together are called pyrophosphate. I don't know why pyro. Might be a Greek word. I'm not really sure. And then it's going to add on to that 3' end. So remember, the growth of the new strand is 5' to 3', just like that. And so on until it basically gets to the end of the strand, or you've got the merging of two replication bubbles. But the thing about DNA polymerase is it needs a little bit of help. And because there's a lot going on here. So what I want to do is kind of show you a breakdown of DNA replication. And if you take a look at this diagram here, and we're going to get into this a little bit, but there's a few different strands going on. So the origin replication, for starters, is right here. So let's think about there. You've got the DNA separate. And then let's just focus here on the first strand here, this leading strand. You can see the DNA, it replicates, and it's going along, and it's adding new base pairs in the complementary fashion, growing in the 5' to 3' direction, and so on. And so right over here, the replication fork is going to keep opening up, and that's going to go on and on and on until it gets to the end. This other thing here, the lagging strand, is a little bit harder to explain, but if you take a look, just take a look over here on the right. So if you can imagine our origin replication, so here's my origin replication, right? And so it opens up, and the replication can only go 5' to 3'. So it opens up a little bit, and we can make a little strand, and this is 5' over here, and this is 3' here. And then it opens up a little bit more, and so we got to make another little fragment, 5' to 3'. And then it opens up a little bit more, and so on and so on. So the lagging strand is done in fragments. These are actually called Okazaki fragments, named after a Japanese guy. And what we're going to do, I'll get into that in a bit, but let's just focus on that leading strand. I want to show you what is going on there, because DNA polymerase can't do it on its own. It does need a little bit of help. So the help part. By the way, there are 6 enzymes and proteins involved in DNA replication. So DNA polymerase is 1. Here's 4 more. And there will be a question on the final exam on them. Last year, I believe it was a matching question where it had the name on one side and the description on the other side. Sometimes I've had it part of the written section, but you can be guaranteed there will be a 6-mark question on the final exam on these DNA replication enzymes and proteins. By the way, you should probably take notes. For the next couple of weeks in class, I'm going to be telling you a lot of things that are on the final exam. And they're not hints, they're going to be promises. So this is my promise to you. There will be a question on DNA replication. A 6-mark question. I haven't decided what format yet, but it's going to be on these different proteins and enzymes. So it's in your best interest to learn about them. Okay, so let's talk about these just for a minute here, and then I'll show you the whole process. First one to note here is helicase, and I will make some actual notes on these. What we're going to do, if we have time today, is I'm going to draw a DNA replication fork, and I'll make some notes on all of these things. So the first one is helicase. So helicase is kind of like an unzipping enzyme. It breaks the hydrogen bonds to the DNA and separates them. So if I were to name it, I'd maybe call it unzippase or something like that, but that's what it does. So it separates the hydrogen bonds. And the hydrogen bonds, they want to reform, right? So, so this is a problem. So what we have here are these things here, these single-stranded binding proteins. So I'm going to abbreviate them as SSBP. And they basically hold it so that it doesn't reform the double helix, holds it in a single-stranded formation. So the other helper that you can see there is this one here, topoisomerase. Sometimes it's also called gyrase. There are a few different versions of this one here. And this enzyme is an untangler. So if you think about it, if I had a rope and I just tried to pull it apart, or if you had your hair in a braid and you just tried to pull it apart, it's going to get tangled. And so DNA is in a helix, and if you just try to separate things, it gets tangled. So this is an untangling enzyme. If you take a look carefully, what it does is it basically puts a little nick in the DNA, so it actually breaks one of the strands, lets it unwind a little bit, and then reforms the covalent bond there. And that's really important so that the DNA doesn't get entangled as it unwinds. Okay, and then the last helper here is this one here, primase. Sometimes I've heard this one also called RNA primase, but our textbook is calling it primase, so we will stick with that. And so what does this do? It's adding the primer. So the RNA primer is shown right here in red. This is just a little chunk of RNA. I think it's about 15, maybe 20 base pairs. I can't remember. It's just a little chunk of RNA. And what is it doing? It's giving this 3' end, so DNA polymerase can get started, right? Because DNA polymerase can't get started without a little bit of help, and it needs that 3' end because that's one of its substrates. As an enzyme, it needs a 3' end, 3' OH. Okay, so let's take a look at the process here, and then, like I said, I'm hoping to have time to kind of draw out the replication fork. Okay, so take a look at this. DNA replication has gotten started, right? So we've got our helicase here, so we'll put an H for helicase. We've got our single-stranded binding proteins here. Our topoisomerase is probably over here somewhere, maybe it's further upstream. You've got your RNA primer, has laid down a little bit of RNA, and then DNA polymerase is in there. So there's DNA polymerase 3. Ignore that sliding clamp. If you want to learn about the sliding clamp, that's fine. And so notice it's adding new nucleotides onto the 3' end, and there it goes. So they're going to be complementary, so A pairs with T, G pairs with C, and so on. And so you can see it's moving along and making more DNA. And that's pretty much the leading strand. So once things get going, you've got all those helpers that separate the DNA and all that, and the RNA primer that allows the DNA polymerase to get started. After that, it's all good, right? It can kind of zip along until it ends up at the end of the chromosome, and or it meets the next replication bubble, and then its job is done. So let's take a quick look here at. This is just some other pictures from, I believe, an older version of the textbook. It's also the leading strand. Sometimes it's just useful to show you a couple of different diagrams, but just showing you one more time, right? So there's the RNA primer right there. The DNA polymerase comes along, grabs on, and starts adding the new nucleotides. And the synthesis, don't forget, is in the 5' to 3' direction. And there it goes. It's zipping along. And so the last thing to mention, as I mentioned, that proofreading activity is, so notice this thing here, this RNA primer. We want DNA, we don't want RNA, right? So what do we do about that? Well, the DNA polymerase, it's actually a different DNA polymerase, but don't worry about that detail. But a DNA polymerase comes along and says, hey, it's proofreading, and realizing that that's not what it wants, right? It's kind of like you write something in pencil, and then you decide you want to have it in permanent ink afterwards. And so you need to come along with an eraser. And so the DNA polymerase comes along and says, that's not DNA. It proofreads, it cuts out the RNA primer and adds the new DNA. And that's what's going on right there. So DNA polymerase has a lot of roles. And then there you go. Now we have our new daughter strand of DNA, at least the leading strand. Okay. So let's talk about that lagging strand a little bit here and what is going on. Much of the process is exactly the same. Like I said, the problem is it can only be done in 5' to 3' direction. So it has to do it in these little fragments along the way. So let's take a look at this. So here's the same first step, right? You've got a primase here, adding some RNA, and just ignore this other little primer just for a moment, okay? But that's another Okazaki fragment there. And then your DNA polymerase, here it is. DNA polymerase comes along, adds some DNA, and it's moving along. And remember, normally that's going to go all the way to the end of the strand. or until it meets a bubble or something like that. In this case, though, it actually bumps into the next Okazaki fragment right there. So there we go. So it detaches. It can't finish. It doesn't have the ability to attach on to that other end, and it's RNA anyway. It doesn't want to attach to RNA. Okay, so then what happens? Well, as we've said before, we got to keep this going. So you can see another strand is going to be produced right here. So don't worry, we'll get that RNA out here in a minute. But you can see that's actually what's going on right here, is that this DNA polymerase is getting rid of this RNA right here, just like what we did before. That proofreading, it sees RNA, it removes RNA, and then attaches DNA there. So there's one more thing to show you here. So there it is. Again, we've got the DNA polymerase removing the RNA. So one thing that happens is that the DNA polymerase, it's kind of not really shown here, but can only attach to that 3' end. So unfortunately, it cannot attach right here to the 5' end. It doesn't do that. That's not what this enzyme does. So there's going to be a little nick when that whole thing is done. And you can see the nick is shown right here on the bottom diagram. And so a little break. So we need one more enzyme that's going to finish the job. And this is called DNA ligase. So ligase is a chemistry term meaning binding together. And that's what it does. It binds those little Okazaki fragments together at the very end. And that's the lagging strand. Okay, so let me see here what time it is. I think there's enough time for me to try to draw the replication fork. So I'll do that here in a moment. There's kind of a summary of everything right there. And so like I said, I want you to make sure you learn the functions of all 6 of these enzymes and proteins in the process of replication. Okay, there's my reminder to draw the replication fork on the board. So what I'm going to do is I have a virtual whiteboard here, and I'm going to switch to that, and I'm going to draw this whole thing from scratch, and then we're going to write some notes as we go along. So let's see if I can remember how to use this virtual whiteboard. And so you're probably going to need a full page of paper, maybe 2 for this. If you have some handy, that would be great. So let's start off with what I will do is, how do I change the color of this? Okay, we're going to go with, I think that looks like a dark blue. So we'll be consistent with the textbook there. So what I'm going to do is I'm going to draw a big replication fork. It's going to look something like this. Okay, and there it is. All right, and so I'm just going to join these together like that. And so what I'm going to do is these 2 strands are going to have a 5'. Let me just zoom in that a little bit better. I'll probably do a little bit of zooming in and out as I'm trying to draw this. This is on a little laptop screen, so it is what it is. So each strand has a 5' and a 3' direction. So I'm going to draw arrows to show you which direction they go in. Remember, we're always going to the 3' direction. And there's the 5' direction. Oh, that's a terrible 5. Let me see if I can erase that. There we go. Okay, there's a 5. That's a much better 5. Okay, so there is my DNA replication fork. So let's that will be my title way up here. DNA replication fork. Okay, there we go. All right, there it is, my DNA replication fork. Okay, so what I want to do is basically go through the process of DNA replication and kind of draw it and write some notes as we go along. Okay, so what we're going to do, and there's a reason why I drew it in this particular orientation, because I want everything to go 5' to 3'. So the very first thing to do, remember, is to make our RNA primer. So I'm going to do that here in red, right there. So there's my RNA primer. Okay, and so my RNA primer goes in the 5' to the 3' direction, just like that. It's going to be complementary as well. And okay, so let's just put a label on that. Let's see if I can get a black for a label. There we go. So this is my RNA primer. There we go. Okay, so the next thing is going to happen, let me see if I can get a lighter blue color for my new DNA, is the DNA polymerase is going to attach onto that primer and it's going to make new DNA. So there it goes in the 5' to 3' direction. Here we go. Okay, we'll find a new color here. Let's go with, I'll go with green here on this one. So I'm going to draw my DNA polymerase. There's my DNA polymerase right there. Okay. And what I'm going to do is I'm going to write a note about it right there. So I think I can type that in. So this here is my DNA polymerase. Okay, so remember we had actually 6 notes about that. I'm just going to make this one very short. So adds new complementary nucleotides at the 3' end. Okay, and I think I can just shrink that a little bit. Like that. There we go. So there's my DNA polymerase. There we go. Okay, so there we go. So that's the easy part. But maybe the thing I forgot to do is we should have added our helicase and our single-stranded binding proteins. So let's do all that right now. So what I'm going to do is I'm going to add my helicase. Maybe I'll make it a round ball like that. Okay. And my single-stranded binding proteins, I'll make them the little balls like that, and I'll label and put a note here in a moment. So 1, 2, 3. Okay, so helicase, we'll do that one first. Right there. Okay, so this is helicase. Helicase, okay, so separates the 2 strands of DNA. And we can say, if you want, you can say by breaking hydrogen bonds. I figure that's actually a good note to add. I'll shrink that down to size a little bit. There we go. And the single-stranded binding proteins, I think maybe I can put that note at the bottom. How about? So there we go. We'll stick it right there. Okay, let's try that again. There we go. I think I just need to click on it with my mouse, not my pencil. So single-stranded binding proteins, single-stranded binding proteins. So what do? They bind the single-stranded DNA, so that's single-stranded DNA there, and prevents double helix reforming. There we go. I'll just shrink that down to size a little bit. There we go. That looks a little bit better. Maybe I'll keep it like that, because what I want to do is I want to add in another feature here. So let's see here. I'll try a new color. How about for this one? The other one we need to add, I'll make it a different shape. I'll make it more of a diamond shape like that. And this is my topoisomerase. Uh-oh, what happened to my note on my single-stranded binding proteins? Oh no, I'm going to have to write that one again. Let's do that again. Sorry about that. So SSBP, single-stranded binding proteins. It's not fun having to retype, but whatever. Binds single-stranded DNA and prevents double helix reforming. Okay, and we shrink that down and move it right there somehow. I don't know how to move it. It's got to be a way to move it, but I don't know how. So let us label that topoisomerase. I'll put the label over here somewhere, I guess. Topoisomerase. So this untangles the DNA. So untangles and reduces the tension in the unwinding DNA strand. Okay. That note, there's got to be a way to move these notes around, but there we go. I will draw that and go like that. Right there, topoisomerase. Okay, so we have we have a lot of proteins there, right? We haven't put that's 4 of them. Remember, there's 6 of them. So maybe a couple of other notes to put on. So in case you haven't figured this out, I'm going to put a big arrow here, right? So this here arrow, basically, I'll type this. My typing is neater than my writing. So this is the direction of replication. So there we go. We got that there. Oh, I don't know how I moved that before. How did I do it? Okay, well, I guess I'm not moving it. Okay, so we want to dissect the lagging strand, and then that will wrap up today's lecture. So let's take a look at the lagging strand, and this one I'm going to zoom right in so that I can write carefully. Okay, and what actually happens, and I'll come back to this in a second. We're going to zoom right in. Is this strand over here is made first. So let me just get the right pen color. I need. Uh-oh, did I just? No, never mind. Okay, there we go. The right pen color. So these little Okazaki fragments, this one here is made first, by the way. So this one here. And then we have another one made. Okay, so these are all made in the 5, oops, that should be a 5. Let me erase that. There we go. These are all made in the 5' to 3' direction, by the way. Okay, and then you have another one made over here. And don't forget that in order to make these, we also have to make our RNA primers. So there's my RNA primer right there. Okay, so this is my RNA primer. And so the RNA primer was made by primase. So maybe what I should have done, where can I stick a primase? Why don't I just tuck a primase right in there? Sorry that it's a little bit small, but I forgot to squeeze that in. So this is primase. And let's write a note for that. So primase. So adds a short segment of complementary RNA as a starting point. Sorry, let me just, there we go. We can zoom out on that for a minute. As a starting point. So remember, that means that we need a free 3' OH and for DNA polymerase. And that's what primase does. Sorry, I had to squeeze it in there really tight. If you take a look at this whole thing, and you probably know because we've been doing this, we've been labeling this already. Let's see here, just get some. This is going to be 5' over here. All these have 5' and 3' ends, so you can label them if you like. Back to green, right here, we've got another DNA polymerase. So DNA polymerase over here. And then the last enzyme, let's find a new color for that one. Let's go with orange for that one. The last enzyme, and we'll do that one as a circle, is of course our ligase. So remember what our ligase does. So our ligase, this one joins the Okazaki fragments together. So this is a covalent bond, of course, the 2 Okazaki fragments together. There we go. Okay, so there is my DNA replication fork. I've got 2 more labels and then we're done. So sorry if I'm just taking 1 minute over time here, but I just want to finish labeling this. And so this here is my leading strand. And this here is my lagging strand. Okay, so there's my DNA replication fork. We will come back and review this after the reading week. I do want to show you one more quick thing, and then I'll let you go for today. And make sure you read your email for today's lab. So I just want to show you this here. This is the DNA replication fork I made last year on the board. So maybe it's a little neater, it's a little easier writing on a real whiteboard than a virtual whiteboard. So if you find this helpful, it is found in the PowerPoints. Other than that, thanks for coming to my Zoom lecture. I will see you after the reading week, and please do keep up on the lab exercises and take a look at the email that I sent yesterday regarding the lab exercises. Other than that, hope you have a wonderful day and a good long weekend.